Hi everybody, so I'm Laura and today together we're going to learn how to navigate through healthcare data. Big topic. So as a bit of context, I'm a senior ML engineer at Owkin. So previously I worked on a medical device system with General Electric, deploying in the hospitals medical devices with AI algorithm in them. And right now I'm working at Owkin's on drug discovery pipelines, but more recently on a copilot focused on helping biology and life science researchers answer complex questions. And I'm also a volunteer data scientist and board member of a non-profit organization called Aura. We want to build an open source epilepsy seizure detection algorithm, as well as along with an open hardware patch, cardiac and movement patch. So we're looking for volunteers. So if you're interested in this mission, don't hesitate to hit me up after this talk. So together I'm going to explain why healthcare data is so challenging. And I will going to present two tools that we use in our copilot that are used to retrieve scientific data. And along with this, I'm going to share a bit the challenges and the lesson we learned along the way. So why is healthcare so specific? So it's a specific domain which has to comply with strict regulation and laws for patient safety, of course. To give you a rough idea how heavy the processes are, I can give you some costs. How much does it cost to have, let's say, a liver segmentation algorithm deployed in a medical device in a hospital and used in diagnosis? It will minimum cost $500,000. And if you want to create a medical device, a computer-aided diagnosis or a computer-aided detection device, it will cost $3 or $4 million minimum because you have to have a mandatory clinical trial. And so, of course, there are software engineer here or data scientists. Imagine having to look through all the open bugs of all your software dependencies and ensure that if they are still open, they do not alter your software, thus the diagnosis. So, of course, ethical question. To retrieve data, patient data, you have to have their written consent. Think about as well the cost. How much does it cost to get an MRI images or a CT scan? And in biology, let's say RNA transcript from a biopsy, from patient biopsy. It costs a lot. The domain is also semantically specific. For example, you have a gene named TP53. TP53 is also the name of the RNA transcript, and P53 is the name of the product the protein is creating, the protein which is coded by the gene. You have BRCA, which are acronyms for breast cancer. It's also the name of two genes, BRCA1 and BRCA2, which can have other names in other semantic or other classification. And so, of course, maybe you attended some yesterday talks about explainability. Here it's even more important because doctors will never trust a black box software algorithm, and even more insurance company which want accountability, they will refuse to pay for any errors that the software did. And really important as well, patient data is sensitive, and every time you use patient data for your algorithm or your software, it has to comply with local laws, of course GDPR or HIPAA in the US. And as a side note, in China, if you want to develop an AI algorithm and have it marked by the Chinese label, you have to use Chinese data, which are only accessible if you develop the algorithm in China. So here's a bit the landscape in biology specifically. There are a lot of open source database out there, ranging from pathology, genomics, biochemistry, To give you an example, I will look at TCGA on the top left. TCGA is a really famous open source patient omics data. You will find for 33 different diseases a roughly 10,000 patient biopsy. Those biopsies have been, there has been an RNA transcript on it, so you have information at roughly 20K genes, protein-coded genes, but other database you can have almost 80K genes. And you have the RNA transcript for every group of cells. For spatial transcriptomics in Mosaic, for example, which are our own database that we offer a subset online, you will find this RNA transcript for every spatial cell in the slide in the biopsy. And along with that, you have the image, which is high resolution image, 0.5 microns per pixel. So it's a lot of data. And so you have three kinds of people working in this area. You have biomeds and wet lab researchers, which know pretty well what to find in the data, but they use Excels and third-party tools. You have biostatisticians that can model pretty well the data, but they code in R and they use notebooks. And you have data scientists. They know how to build tools around this, but they have trouble to scale. And so you need tools for non-programmers, but also that can scale, because they are facing information overload. As you saw in the previous slide, there are a lot of data out there, really heterogeneous, indexed, not indexed the same, with different formats. Some are Parquet, CSV, other are TXT files, and they change constantly, every quarter, every year, and so on. So what we did at Owkin, we developed in 4 months Copilot, which offers for now 3 tools. One that helps you visualize patient data and visualize omics data. I will not talk about this, but I will talk about the 2 other tools. One that enables the researcher looking at the literature, and the other one look at a broader database, a prior knowledge with information on genes and proteins. So, first of all, this prior knowledge database. So historically, at Owkin, we have curated around 17 open source database. We have cleaned it, automized the cleaning, applied biomeds rules that they want to look, the data that they want to look at, so discarded all the unusable information. And it enables them to answer questions like, where is KRAS expressed in the cell? So KRAS is the name of a gene, and we want to look where the protein that is coded by this gene is found in the cell. And you can usually answer it by looking at UniProt database, and they have several columns in it that helps you answer the question. So what they were doing, they were looking at a big Excel file, more than 200 columns, 80K rows, and they're trying to answer the question like this. We wanted to build a tool that would do this automatically from a natural language question and reply with the formatted answer with the website, the link to the database where they can check all the claims that it said. And so we built a tool. So the data, this Parquet file is loaded with Polars, and Polars enables you to query, to filter the data with SQL language, and we wanted something to transform this sentence into a computer-readable language, so that it returns a smaller subset of the database, the dataset, and go to another LLM to create a structured answer, so the answer in free text along with the sources, so that it's given to the Copilot, which will create a more formatted answer, something with the links at the right place and something readable for the researcher. So you cannot put the whole dataset, the whole table into an LLM in the prompt. It will not work, even though now Claude Sonnet 4 has 1 million tokens as input. It will cost you a lot of money, but it's also really slow. So you need to be careful what you put in the prompt. So what we did is that we only put the needed metadata, the schema of the columns. But more than simple database schema, we realized quickly that it had to be really well written in non-ambiguous terms because of the specificity. We have to embed some knowledge on, let's say, the acronyms that I taught you before. What is a gene? What is a protein? What is the relation between them? And also you have to prompt the LLM to be able to create a SQL query, which is going to be read by pollers, to be as simple as possible, to return something that is going to be used to answer the question. But then we realized that the LLM was outputting more than the SQL query. And so it highlighted the need of having structured output. So we used PyData and Instructor, which is a tool which is going to force the LLM to enforce a schema defined by PyData. And if it doesn't succeed, it will retry internally. But you can use more advanced tool at the token level, which will mask on the inference the tokens that are not in your schema that you define, tools like outlines or JSON former. Also, you have to ensure that this SQL query which has been created is valid and not malicious for your data. What you can do is blacklist, whitelist important keywords like update, erase, and so on. But you can also launch your SQL query in a sandbox environment with not your production data to ensure that it's valid and not malicious. And also Polars has it will raise exception if for example, the SQL is not valid or too complex. Also, you have to be careful with which the output of the subtable you get, because it's going to be the input of another LLM. And if it's too big, you will reach a max token, or it will cost you a lot. So what you usually do is that you summarize, if it's a text, for example, summarize, semantically aware. But here, if it reaches a certain number of rows, we use describe, polar describe, to give only meaningful statistic about the table. And so also from a cost point of view, but also accuracy, on the first LLM here, if you give the whole description of the thorough description of all the columns, you will reach a problem called lost in the middle. The LLM will have so many information that it will not know how to use it to answer the specific question, but also it's going to cost a lot. So we refine iteratively to only give at the end the needed columns to answer the specific question. So here is a bit a summary of everything that we encountered and what we put in place along 4 important topics: accuracy, security, cost, and latency. And I want to emphasize, to highlight something that we discovered as well early on, is that everything you put in the LLM prompt, you have to be comfortable sharing it with the public, because it's really easy to retrieve a system prompt by a smart query. There are today some tools that exist, some guardrails that will check your prompt for any malicious or anything like this, but it adds latency to your system. And even more here, you don't want patient data to be outputted and visible. Then we will look to this tool that will help the researcher getting information from existing literature. In life science and biology, they use PubMed, which is one of the biggest platforms where you can find more than 26 million of abstracts. You have, it's regularly updated, and you have, every year you have roughly 1 million abstracts updated, and also discarded. So we use a really famous technique called RAG that you probably already know. So it just enables the LLM to not only relies on its pre-trained data, but on your own data, your own documents, here the abstracts, and give a more accurate answer with the possibility to give the sources. So usually you encode your documents, here the abstract, in a space where items will be, if they are semantically different from each other, they will be really far away in this space, and if they are close semantically, they will be really close. So when you have a question like what is the role of plur in cancer, you will encode this question and retrieve the, let's say, top 100 items around the space of the closest, and you will give them along with the question and ask another LLM to answer the question with this information. So here we encoded 26 million. It costs a lot, and also it takes a lot of time, 24 hours to ingest. And we internally ask researchers to ask this question: what is the role of plur in cancer? Plur is the name of a gene. It encodes a protein called UPAR. And researchers were like, okay, but it gives me back some papers about LY6 plur. So like, okay, there's plur in the name, but it happened that it has nothing to do. LY6 plur is also called LY6 UPAR, and it's the name of the family, of a domain of a protein family. So, which has UPAR, which UPAR has in common with other proteins. So it should not retrieve this kind of paper. And so we decided to go with an hybrid search. So instead of relying only with the keywords, which are encoded with sparse vectors, you're going to use it along with dense vectors, which captures more the semantic of the sentence. And those together will give you better results. Then the researcher said, okay, it only brings back, it brings back reviewing papers. So in scientific community, you have papers that talks about other papers. Those are, okay, interesting, but you want the original paper. So what you can do to fix this is to have index on your vectors, where you're going to have metadata, let's say author, a date, and here if it's a reviewing or original paper, to be able to filter before the paper you really do not want to get. You can also add a re-ranker after the retrieval of those, let's say, top 100, to be able to ask what are the papers that are the most important to answer my answer. Here's a bit, again, a summary along those 4 important things. Something that I want to highlight is that working with vector database, which are just vectors with numbers, is also working with patient data, if there is, or if you're encoding patient data. So it has to comply with GDPR and or HIPAA. Something to think about. So, as conclusion, something we realized really quickly is that we started developing in January to have a launch on May, and things are really going really fast. You choose a framework, you choose an API, you choose a model. Maybe tomorrow it's not the best model. Here we used Anthropic, and here it's not the best model here. Also different framework that would do everything, but are not really stable. And so you have to think about it when you develop your product. Also, here science is hard to evaluate. Usually biology question cannot be answered by yes or no. It's almost all the time, it depends. So it depends of what? It depends of a lot of things. And something that has been highlighted recently is that, I don't know if you know the Humanity Last Exam, which is a benchmark where all the, on which all the most famous models are benchmarking their model on. And the biology, life science part of Humanity Last Exam, they found that there were almost 30% of the Q&A in it that were probably wrong because of non-consensus on some question, and sometimes the answer was not detailed enough to really make a decision on it. So if I have something to say is that if make your own benchmark with domain experts, validate your tools and your agents to have to really control what your tool is doing. And here at Owkin, we are building this copilot not to replace researcher, but we want to help them look at the data, the open source data that is available, but also proprietary data. We offer a subsample of spatial transcriptomic data from Mosaic that they can look at, and soon we would like to offer the possibility that they add their own data to the analysis. And I think I'm a bit early on.
Thank you, Laura. So we have a couple of questions in the app. Okay, I'm going to start with the old one. So I'm reading as it is, okay. Can text to SQL translation handle more complex queries like aggregations or joins?
No, we realized that, so that's why maybe I forgot to say it, but it has to, when you do this natural language to SQL, it has to be simple queries. And we realized early on that nested or we weren't doing joins because we weren't having more than one table. But I think this is something challenging when you have multiple tables, so when you work with the big and a lot of table in your database. But the key is really the context, the domain context that has to be really clear. And also Polars doesn't handle all the SQL queries.
Thank you. Any question here in the room? Please raise your hand. And yeah, there is one. Yes, there's another one. Yeah, just here.
Hi, I was wondering, so you were looking for instance, for scientific papers with the examples were very short phrases, right? So if semantically maybe you get 1,000 papers where like a phrase like this occurs, what kind of metric can you use in order to ensure that the outcome is most relevant for the search?
So when you have a long query, it's good to divide it in like more than one query, to more than one subject, and then to look for the subject and then gather the outputs at the end. Re-rank with the complete question, but we haven't tried this because maybe I didn't say during the call is that we spent some time and money developing this RAG, and a few weeks later, PubMed did its PubMed AI, which of course works really well, and because it works with the entire paper. And with abstracts, we really hit a wall on the information that is given in the abstract.
Thank you. There is another question with a couple of votes here. I'm going to read this one. What's the current daily usage and the main user groups of the system? How did the actual usage in the production differ from the initial design and expectation?
So how did we monitor? What are the usage and which type of people? So first, when we released the product, it was only meant for a biology researcher, academics, and we wanted to do it for the whole medicine community. But in France, you have a regulation. It's called the gift law. You cannot offer a tool for free to doctors if you're a biopharma, and which we are, because we have a molecule in clinical trial going on, and so that you cannot give away. So it was really focused on academics and researchers with no link, so no medical students as well, no link with the healthcare industry. And for the monitoring part, so we used to monitor lung fuse and open telemetry. And we add also on the copilot human feedback, the availability to give thumbs up or thumb down, and add a review on what's going on if it's bad. And we are still processing the outputs of this, of the production feedbacks. But the good thing at Owkin is that we have a large panel of internal users, biomeds, biostatisticians, and data scientists that can really help us get feedbacks pretty soon.
Thank you. So another question. Does the RAG system consider the accountability of different scientific journals? Is there a scoring system to be able to give certain weights to deliver best possible answer based on journal quality?
Indeed. So what I would think about if you encode multiple journals in one RAG is to add the name of the paper and maybe give a note, a weight to its relevance as index of the vectors that you encode. And then on your system, prompt to do it in the re-ranker to think about the provenance when answering the question and giving the citation to the claim in the answer. Yeah, that's what I would think about.
Thank you. So another question. Does abstract means only the summary of a paper? Do you have access to the full paper text?
No, unfortunately, on PubMed, there's only abstracts, 26 million abstracts, and 10 million full papers. So you have to pay to get the full paper. And also, encoding a whole paper costs a lot. So roughly, we use Quadrant to host our vector database. It costs us around $800 per month. But you have to think as well about the hidden costs. So the creation of the embeddings, which are really costly in computing, and also the maintenance of the database, because as I said, some papers are added every month, every day, but some of them are discarded because they are not relevant anymore. So, yeah.
Thank you. So another one. Have you tried using schema or metadata of the database in the system prompt of the LLM?
To what, sorry?
Yeah, have you tried using the schema or metadata of the database in the system prompt of the LLM? So,
data schema gives you an idea of the name of the column, its type, and so the metadata that we actually do is this, plus really thorough explanation of what is the column. Because if you have a column named localization cytoplasm, and you don't give any context other than it's a Boolean, or if it's categorical data, it's like, I don't know, ABC. If you don't give any context to this, then it will not help the LLM get an answer.
Thank you. So we have time for one more question. What's the challenge to use a proprietary LLM with sensible data? If yes, what are the approaches to overcome this kind of data?
So for this tool particularly, we used open data, so they are pseudonymized, and usually, well, patients are dead. That's why it's so easy to have their data online. And so there were less constraints using all this data for the other tool, for example, that enables to visualize the histomic slide along with the features, histomic features, and so on. I would say, from my point of view, the challenge was, as a developer working on local or development environments, you're not permitted to have the data, the real data that we're working on, even though it's pseudo-anonymized. And so you have to get synthetic data, but it's not easy to make synthetic data of omics data. It has to have a meaning. You cannot put random values. It has to coincide with other data to, let's say, do a differential gene expression. So I think from my point of view, that was the hardest. And I think we don't plan to have this as a medical device, so yeah, I don't know.
Okay, thank you very much, Laura, and please.